lipid metabolism, and we will cover all of the highlights of this chapter 17 in this one lecture because our time is short. So to begin with, I wanted to remind you of triacylglycerol structure. So triacylglycerols are just like their name suggests. Glycerol is shown here in black. And if we esterify those three OH groups on glycerol with a carboxylic acid, which are the fatty acids, then we get these ester functional groups. Remember, acyl means ester to a biochemist. So these are triacylglycerols. So glycerol is the backbone, and then three fatty acids that form these three ester groups. So these triacylglycerols, remember we talked about in chapter 9, they are completely nonpolar and they're stored in an anhydrous form because if you're nonpolar, you don't like water. In contrast, glycogen storage is limited. Fat storage is not limited, as we can see with the obesity problem in this country. Glycogen storage in the muscle and in the liver is limited, and 1 gram of glycogen binds 2 grams of water. So that's partly the reason for some of these low carbohydrate diets. What you end up doing is depleting your glycogen, so you lose 1 gram of glycogen and you lose 2 grams of water along with that. So that accounts in part for some of the weight loss that you see with those diets. So I thought you might be interested in this figure right here. When you're doing different kinds of exercise, so something that just takes a couple seconds, like high jump, power lift, shot put, tennis serve, we use ATP. But if it goes above, say, 3 or 4 seconds, you'll need phosphocreatine, and phosphocreatine can donate a phosphate group to ADP to form ATP. So you can get a little extra ATP that way. If the duration is only a few minutes, you can go through glycolysis and you can do this anaerobically to get 2 ATPs per mole of glucose. But if you have some long duration kinds of exercise, you'll have to undergo oxidative phosphorylation. And we talked about how glucose can be degraded to CO2 in water using oxidative phosphorylation, and you get 30 to 32 ATPs, so you get a lot more ATPs, and you do need oxygen for that. You can break down fats using oxidative phosphorylation as well. And in fact, with fats, there is no choice. If you're going to break down fats, you need to have oxygen. So in the where are we section, we are here. So we are going to take lipids, specifically triacylglycerols, and then we're going to hydrolyze those acyl groups to get 3 fatty acids, and we'll take them through a pathway called beta oxidation, and we'll produce acetyl-CoA. And then acetyl-CoA can go into the citric acid cycle and produce NADH and QH2 for electron transport. So why this is called beta oxidation, we'll get to in a second. And we should note that you get a lot more energy from fat breakdown than from glucose breakdown. So this figure, I think, shows a little bit too much information. So after we go through this whole PowerPoint, you might want to go back and look at the figure, because I think here there's just maybe a little bit too much going on. But here we note that fatty acid degradation, or this beta oxidation pathway, will occur during fasting. And during fasting, the pancreas is producing glucagon. And glucagon can act on the adipose tissue and put into motion a series of events that leads to the lipolysis of triacylglycerol to fatty acids. And then the fatty acids can leave, and then they can go to other body tissues that need it. Glucagon also acts on the liver in carbohydrate metabolism, as we talked about earlier in the course. Epinephrine is also released during fasting, and again, earlier we talked about how epinephrine acts on different receptors, but also collaborates with glucagon to increase the degradation of glycogen and gluconeogenesis. During exercise, epinephrine acts on the adrenergic receptors in muscle to break down glycogen to glucose 1-phosphate, which is isomerized to glucose 6-phosphate, and then enters glycolysis. So we will focus our efforts on the adipose tissue for now. So how do we begin? Well, we did begin by taking the triacylglycerol and hydrolyzing those 3 fatty acids to glycerol and the 3 fatty acids. So this would be one of those fatty acids here. So this fatty acid right here, before it enters the beta oxidation pathway, has to be activated. It's sort of like glycolysis when we had to spend 2 ATPs to get 4 ATPs back. Well, this is sort of similar. So we'll need ATP, and ATP will go through an intermediate where we have an AMP attached to our fatty acid, but then that is hydrolyzed to give us an acyl-CoA. So we have coenzyme A, and now there's our fatty acid shown there in red. So we'll have a fatty acyl-CoA. So it's going to cost us the hydrolysis of 2 phosphoanhydrate bonds to do that. So we'll do a hydrolysis here, and we'll release pyrophosphate, and then we'll hydrolyze another phosphoanhydrate bond to yield 2 inorganic phosphates. So, and then again, AMP is our leaving group. So what we've done in this step is we have spent 2 phosphoanhydrate bonds to synthesize acyl-CoA. So we do need 2 phosphoanhydrate bonds, so the hydrolysis of this pyrophosphate right here drives the reaction to completion. So what happens next? Well, the fatty acyl-CoAs are generated in the cytosol, so that's where we are down here. But beta oxidation takes place in the mitochondrial matrix, so that means we have to get the fatty acyl-CoAs from the cytosol into the mitochondrial matrix. So the carnitine transporter is going to help us out. It turns out, though, that you can't just take fatty acyl-CoA and transport it across. What you have to do is you have to trade the coenzyme A for a carnitine. So here's our acyl group, our fatty acyl group right here in red, and now it's attached to carnitine. And now it can go across. And the acyl carnitine is now in the mitochondrial matrix. And then the reaction will happen in reverse. We'll trade back to regular carnitine, and now here is our fatty acyl-CoA, and this carnitine will leave to complete the cycle. And now our fatty acyl-CoA is in the mitochondrial matrix, and we can go on to beta oxidation. I just want you to put in your mind for right now that carnitine will be allosterically inhibited by malonyl-CoA. Now, that doesn't make any sense right now, but it will in a minute, I promise. So let's use this animated figure to describe what's happening. We will need our Adobe Flash Player in order to show this. So here we start with our fatty acyl-CoA, and in the very first step, there is a dehydrogenase, so we know it's a redox reaction, that will take off these 2 hydrogens, and now we have a trans-enoyl-CoA. So those 2 hydrogens and their electrons will be donated to FAD to form FADH2. Okay, what happens next? Well, a hydratase will allow us to use water, and we'll add water across that double bond. And then we'll use another dehydrogenase, and we're going to pull off these 2 red hydrogens right here. And now where we used to have a CH2, now we have a C double bond O. So if you remember the old nomenclature for naming carboxylic acids, you wouldn't start with the carboxylate carbon, you'd start with the first one, and that would be alpha, and then beta. So that carbon that's shown in red here, that's the beta carbon, and it has been oxidized from CH2 to C double bond O. So that carbon's been oxidized. So that's why this is called beta oxidation. So again, these 2 protons and electrons were donated to NAD+ to form NADH. And the next thing is, here's a molecule of coenzyme A, So we're going to perform not a hydrolysis, not a phosphorylasis, but a thiolysis, where we're going to break this bond sort of with sulfur. And what we're going to do is if we break this bond right here, but if we bring in coenzyme A, then we will have acetyl-CoA. So what we're doing is we're clipping off these 2 carbons of our fatty acyl group and attaching coenzyme A to it. So now we have acetyl-CoA. And now our fatty acyl-CoA is 2 carbons shorter than it was. So that means the next thing we can do is we can start the cycle all over again and go to the first step where we take those next 2 carbons and go ahead and take off the hydrogens and electrons, donating them to FAD to form FADH2. So this video doesn't show the rest of that, but it does show what happens to the FADH2. Remember where we are. We're in the mitochondrial matrix. So the FADH2 can give its protons and electrons to complex 2, that's what this is right here, and then to coenzyme Q to form QH2, and then QH2 then goes through the rest of electron transport. Now it's okay, and they're going to continue this. So you can tell this is an old video because they show that for every QH2 that goes through the electron transport chain to oxygen to form water, you form 2 ATPs. Remember, there's a little proton leakage, so it's really 1.5 ATP. So you can tell this is an old animation. But what it doesn't show, if I go back to my PowerPoint, is that remember, we formed an NADH here as well. So that goes into electron transport too. So I don't know why the video doesn't show it, but it doesn't. So anyway, so every time you go through one cycle of beta oxidation, you clip off 2 carbons to form acetyl-CoA, and you get 1 FADH2 and 1 NADH, and both of those go into the electron transport chain. So the next question I would have asked you this on Top Hat this morning was, how many beta oxidation cycles are required to break down palmitate, which has 16 carbons? The temptation is to say 8, but the answer is 7, and that's illustrated here. And if you look at taking something that's 16 carbons long and clipping them off 2 carbons at a time, think about going down here. So the last cycle is you're going to do a hydrolysis right here, so you get 2 molecules of acetyl-CoA in the last cycle. So it's 7 cycles and not 8. Okay, so now we're in a position to be able to calculate how much ATP we can get. So per cycle, we're going to do 7 cycles. Every cycle gives us a QH2, so we're going to have 7 of those. Every cycle gives us 1 NADH, so we'll have 7 of those. Every cycle gives us 1 acetyl-CoA, except for the last one that gives us 2, and the acetyl-CoA can go into the citric acid cycle as long as OAA is available. So this is what I mentioned before. So since the products of beta oxidation rely on the citric acid cycle, that means fatty acids are oxidized exclusively aerobically. It's not like glucose where you had a choice whether you could oxidize it partially to get 2 ATPs or completely to get 30 or 32. Beta oxidation has to have oxygen. So for our palmitate, here's our balanced equation. We went through 7 cycles of beta oxidation. So there's our 7 FADH2s and the 7 ADHs and the 8 acetyl-CoAs. So when we're calculating the ATP yield, we should know that we do 7 cycles. And another number to sort of keep in your mind is acetyl-CoA for 1 turn gives us 10 ATPs. And the way we get that is that, remember, 1 turn of the citric acid cycle gives us 3 NADHs and 1 FADH2 and 1 GTP. So if we have 1 GTP, that's 1. If we do 1 FADH2, that's 1.5. So now we're up to 2.5. And if we do 3 NADHs, and if each of them gives us 2.5, 2.5, that means 2.5 times 3 is 7.5. So 7.5 plus 1.5 plus 1 equals 10. So 1 acetyl-CoA will give us 10 ATP. So that's a number that's nice to have in your mind. So the other thing we should note is that we get more ATP per carbon. So usually I would ask, well, why is it that we get more ATP from palmitate than we do from glucose? And then the students would say, well, we have more carbons. We have 16 carbons in palmitate, whereas we have only 8 in glucose. And that's part of it, but that's not the whole story. The other thing to note is that the carbons in palmitate are more reduced. They are, almost all of them, except for the first one, are all completely bonded to hydrogen, whereas the carbons in glucose are partially oxidized. So you get a whole lot more of NADH and FADH2 when you break down fatty acids than you do from glucose. So that's why fat is so fattening. So here, if we add everything up, in our citric acid cycle, we get 1 QH2, which is 1.5 ATP, and 1 NADH, so that would be 2.5. So, so we get 1 QH2 and 1 NADH per round of beta oxidation. And then here's our 10 that we get from acetyl-CoA. There's our 7.5 plus 1.5 plus 1. So the total that we get per cycle is 14. So if we did 7 rounds times 14, that would give us 98. And remember, we get that extra acetyl-CoA in the last round. So that would give us 108. But remember, we spent 2 ATPs to activate the fatty acyl-CoA, so we have to subtract that. So our total is 106 ATP for just one palmitate fatty acid. So in our book, there are lots of reactions that describe how we obtain ATP from fatty acids that already have some double bonds. So if you look back in the animation I showed you where there were 2 steps where we did redox reactions where we pulled off hydrogens to yield a double bond. There are isomerase and reductase enzyme that's the kind of, if you encounter a double bond, will sort of move the double bond so it's in the right place. What that means is you don't get quite as much FADH2 and NADH from these doubly bonded fatty acids as you do from the fully saturated fatty acids like palmitate. Now, it is possible to have odd chain fatty acids. Plants make odd chain fatty acids. So that means in that very last step, instead of getting acetyl-CoA, you're going to get 3 carbons. You're going to get propionyl-CoA. So we have this enzyme called propionyl-CoA carboxylase. Hopefully that looks familiar. Remember pyruvate carboxylase, where we used CO2 and we needed biotin to carry the CO2, and it was an uphill reaction, so we needed ATP. So what we did is we add a carboxylate group onto the propionyl-CoA, and we get the S-methylmalonyl-CoA. There's a racemase we need to convert it to the R-methylmalonyl-CoA, and then a mutase that requires vitamin B12, and we convert that to succinyl-CoA. So from succinyl-CoA, that can hop into the citric acid cycle. But here, notice that we started with 3 carbons. Remember, 3 carbons is the magic number that we need to make glucose. So there's actually a little piece of this fatty acid that we could use to make glucose, but not anything from acetyl-CoA, because if you have 2 carbons, you can't get to glucose, at least not in mammals. So I think this is an old edition of the Stryer textbook that had this phrase, fats burn in the flame of carbohydrates. So I like this because it shows how lipid metabolism relies on normal carbohydrate metabolism. So the carbohydrate metabolism I have drawn here in blue. So if glucose is converted to pyruvate and then pyruvate's converted to acetyl-CoA, we have acetyl-CoA for our citric acid cycle. But remember, the pyruvate carboxylase enzyme can convert pyruvate to OAA. And that's normally what's going to happen with carbohydrate metabolism if we have lipid metabolism too, because the lipid metabolism is going to produce the lion's share of the acetyl-CoA. So that provides both of what we need to accomplish that first reaction of the citric acid cycle. Remember, the first reaction, we need OAA plus acetyl-CoA to get the whole thing going. So if we have normal carbohydrate metabolism, that will provide us with as much OAA as we need to process all the acetyl-CoA. Remember, in the citric acid cycle, OAA is regenerated, so you don't need exactly the same amount of OAA as you have acetyl-CoA, but you do need some. So this is what happens if carbohydrate metabolism is not normal, and there could be several reasons for this. It could be maybe untreated diabetes, but this is also what happens in these keto diets that everyone seems to be all excited about these days. So what happens if you're really restricting carbohydrates, then what happens is you'll feed into other dietary components, such as protein will break down to OAA, and you'll get your OAA, but what happens is OAA will go this way, will go to gluconeogenesis instead. So if you're not eating glucose, red blood cells need glucose. The brain needs a ton of glucose. So your body will do what it needs to do to synthesize glucose if you're not eating it. So what that means then is lipid metabolism, which also lipids are also very high in these keto diets. Lipid will be broken down to acetyl-CoA, but now since all of the OAA is diverted to gluconeogenesis, you don't have enough OAA to condense with acetyl-CoA in the first reaction of the citric acid cycle. So we have to do something with that, and what we do is acetyl-CoA forms ketone bodies. So that's why this is called the keto diet, because the blood of the people who go on this diet is very high in ketones. So what do these ketone bodies look like? Well, acetoacetate is a ketone body, and it does have a ketone group. You can actually non-enzymatically remove the CO2 and you get acetone. You know what acetone smells like from being in the lab, and sometimes it's also used for nail polish remover. So what that means is that if you have a lot of acetoacetate in your blood, you'll produce acetone, and some of that crosses from the blood into the alveoli, and you'll breathe it out. So if you go to some of these websites about the keto diets, they'll complain about bad breath. This C double bond O right here can be reduced to beta hydroxybutyrate. So this is not strictly speaking a ketone body because it doesn't have a ketone, but it's called a ketone body anyway. So ketone bodies are produced from excess acetyl-CoA, and ketone bodies are produced in the liver. Okay, that's where they're produced. That's where we have the enzymes to make these ketone bodies. And this is what happens if acetyl-CoA is accumulating because of lipid breakdown and in the liver, which is making undergoing gluconeogenesis and using OAA, you don't have enough OAA to enter the citric acid cycle. So the acetyl-CoA forms ketone bodies instead. So the liver makes the ketone bodies but doesn't have the enzymes to use them. So the liver will release ketone bodies into the blood, and there are other tissues that can use them. They'd rather have glucose, but they can use the ketone bodies if they have to. One of the concerns is that you can see these have carboxylate groups, so they're acidic and they will decrease blood pH. So what happens when the ketone bodies are released into the blood is that other tissues can take them up. So the heart muscle can use ketone bodies. The brain can use ketone bodies. The brain would much rather have glucose, but it can use ketone bodies in a pinch. So what happens is these tissues will take up either beta hydroxybutyrate or the acetoacetate. And then what happens is there are enzymes in these cells that will convert these things back to acetyl-CoA, and then the acetyl-CoA will enter the citric acid cycle, and then those tissues can generate energy from these ketone bodies. So again, it's really important to think about where this is happening. So ketone body formation happens in the liver, and they are exported from the liver. So this sort of tannish thing right here is meant to represent the hepatocyte, which is liver cells. So here shows there are lipid droplets in the liver cells, and they're breaking down lipid to acetyl-CoA. So that's happening here from beta oxidation, which I just showed you. But in order for the acetyl-CoA to enter the citric acid cycle, you need OAA. But if you're fasted, OAA is not doing that. It is being diverted to gluconeogenesis. And remember, the liver allows glucose to leave. So glucose is exported as a fuel for brain and other tissues. So that means the extra acetyl-CoA then forms ketone bodies, the acetoacetate, the beta hydroxybutyrate, and acetone, and they also leave. So heart, skeletal muscle, kidney, and brain are able to use ketone bodies if glucose isn't available. So the liver is doing its best to get glucose out there as well, but the ketone bodies can be used as a source of energy as well. So again, ketone bodies are formed in the liver and exported in the liver, and the liver does not use them itself. It doesn't have the enzymes to use them. It only has the enzymes to make them. Okay, so that is catabolism. Now, so let's switch gears and talk about anabolism. So everything I just told you would be relevant to the fasted state. Well, what about the fed state? So the fed state, we are going to be making fatty acids. So we are here. So acetyl-CoA, we can get acetyl-CoA from glucose or from amino acids, and we can use those to form fatty acids and then lipids, and we can use them to make membranes. And if we have extras, we'll use the fatty acids to make triacylglycerols, and then we'll store it as fat. So acetyl-CoA is fairly highly oxidized, and to make fatty acids, they're fairly well reduced. So we're going to need acetyl-CoA, and we're going to string those together 2 carbons at a time. But we have to reduce these fatty acids too, so we need a source of protons and electrons. So NADPH is going to be our proton and electron donor. All right, so okay, so here is another sort of busy picture. So the picture I showed you earlier was in the fasted state. This is the fed state. So here's insulin. So insulin acts on liver and muscle and adipose tissue. So we've already talked about how insulin acts on the liver to activate the protein phosphatase 1, and that causes glycogen synthesis to be active. Glucose comes in from the portal vein when you're eating, and you can use that to make glycogen as well. Insulin acts on muscle. There's a GLUT4 transporter here, and this insulin is required to be sure that the glucose transporters are displayed on the plasma membrane, so glucose can come in and the muscle can make glycogen as well. And insulin will act on fat cells, and again, it allows glucose to come in through the fat cell and use that as a raw material to make fat, because we can make all of these things when we have the resources to do that when we're fed. So how does this work? Well, I told you that beta oxidation or fatty acid breakdown occurred in the mitochondrial matrix. Synthesis occurs in the cytosol. So that means if we have a whole pile of acetyl-CoA here in the mitochondrial matrix, we have to get it out into the cytosol so we can use it to make fatty acids. So this is how it works. We're going to borrow the first reaction of the citric acid cycle. So we have OAA condenses with acetyl-CoA to form citrate, and then the citrate is able to leave and go into the cytosol, and then the reaction occurs in reverse. So here we have a cytosolic enzyme called ATP citrate lyase, and then we get our acetyl-CoA. So now we can start using that to synthesize fatty acids, but you should also notice that we need to continue the pathway because OAA, again, it would be nice if we could just have this cross, but there's no OAA transporter. So what happens is OAA is converted to malate, and then malic enzyme converts malate to pyruvate, and one of the things you get is NADPH. So remember, NADPH is what you need, so this cycle helps you generate about half of it, and then the other half comes from the pentose phosphate pathway. Remember that? So anyway, we need to complete our transport. So pyruvate, okay, again, so NADPH goes to the fatty acid synthesis, and again, the other half comes from the PPP, and then we continue. Pyruvate is converted to OAA, and that completes our cycle. So all of this is necessary to get acetyl-CoA out into the cytosol so we can use it to make fatty acids, and to get us about half of the NADPH that we need, because that's the proton and electron donor. So you should also notice that since citrate is part of this, that citrate provides a route to fatty acid synthesis. So that's something that, so if citrate levels rise for any reason, we can get fatty acids from that direction as well. Okay, so we've already seen one thing that's different about fatty acid beta oxidation and fatty acid synthesis. So when we break down fatty acids, that happens in the mitochondrial matrix, and when we synthesize them, that happens in the cytosol. Here's another thing that's different. When we break down fatty acids, they are attached to coenzyme A. In fatty acid synthesis, the fatty acids are attached to acyl carrier protein. So notice that the chemistry is the same. We have this phosphopantethene group, and we have the SH right here. So just as we often abbreviate coenzyme A as coenzyme ASH, we can use ACPSH. But this is a whole protein. There's about, I don't know, I think 77 amino acids in this protein, so they don't obviously draw the whole thing. The chemistry is the same, but we'll find acyl carrier protein is used in fatty acid anabolism. Okay, so what else happens? Well, we need to kind of boost our free energy of our acetyl-CoA, so we can't just start stringing a whole bunch of acetyl-CoAs together. We need to activate the acetyl-CoA and form something called malonyl-CoA, and here is again another carboxylase where we have CO2. It's an uphill reaction, so we need ATP. We biotin to carry the CO2, and we convert acetyl-CoA to malonyl-CoA. So that means that the acetyl-CoA carboxylase is a really important reaction. In fact, it's the committing reaction of fatty acid synthesis, and we are going to regulate this by phosphorylation. So this enzyme is active when it's not phosphorylated, and it's inactive when it's phosphorylated. So let's go to this animated figure. How are we going to synthesize our fats? Well, we are going to start here. Here's our acetyl-CoA. But remember, we have to swap the coenzyme A for ACP. So now we have acetyl-ACP. And the next thing that's going to happen is there's this huge enzyme complex called fatty acid synthase. So there are many active parts of this. So what we're going to do is we're going to transfer this acetyl group to the condensyne enzyme that's part of this complex. So we have this, the acetyl-CoA is covalently attached temporarily to the enzyme. Now we're going to bring in our malonyl-CoA. So remember, malonyl-CoA, so the red carbons came from acetyl-CoA and this blue carbon came from CO2. So we're going to have to do the same thing. We're going to have to trade the coenzyme A for ACP. So now we have malonyl-ACP. So now we can carry out our first step, in which we take our malonyl ACP, CO2 is the leaving group, and now we can attach that acetyl group onto the malonyl, what was the malonyl-CoA, and now we have acetoacetyl ACP. So again, these two red carbons came from the malonyl-CoA, that came from the acetyl-CoA, and then these green carbons came from another acetyl-CoA. So now what? Well, remember, we're making a fatty acid, so everything has to be really reduced. So we're going to use NADPH, and we're going to donate 2 protons to the C double bond O. So we have a hydrogen bond here and an OH here. And then we are going to do a dehydration. So we'll take off the 2 hydrogens. So we'll have a double bond here. And then we will need another NADPH to donate protons and electrons. And now we have butyryl ACP. So now we have a short little fatty acid. It's only 4 carbons long. So what we do now is we start all over again. We bring in another malonyl-CoA, and then we add 2 more carbons. So we get 6, 8, 10, 12, 14, 16. So you'll have to do this, those cycles I just showed you 6 more times. And then you'll get palmitoyl ACP. And then there's a thioesterase that does this hydrolysis to release the ACP, and now we have palmitate. So this big fatty acid synthase complex produces palmitate. So palmitate is 16 colon 0, 16 carbons, all reduced, no double bonds. All right, so let's go back to our PowerPoint. So there's our palmitate. That is the product. So what we did is we needed an acetyl-CoA, 7 malonyl-CoAs, but remember those 7 malonyl-CoA came from acetyl-CoA, so we really used 8 acetyl-CoAs. We went through 14 cycles, and each cycle needed 2 NADPHs. So we got our palmitate and 14 NADP+. So the product will be palmitate. So that we can study these reactions in detail, but our time is short. So we'll just simply say for now that if you want longer fatty acids or unsaturated fatty acids, then they're produced from palmitate, and their enzymes elongases and desaturases and the smooth endoplasmic reticulum that can accomplish this. So at the last 35 minutes, summary is right here. So, and it shows both the triacylglycerols being broken down into fatty acids during beta oxidation to give us FAD and FADH2 and NADH for one cycle. Here's our acetyl-CoA, goes through the citric acid cycle if we have OAA, goes to ketone bodies if we don't. And then synthesis is in the up direction. We need NAD, pH, and ATP in order to make the fatty acids, and we can use those to make membrane lipids, or we can store them as triacylglycerols for the next time that we're either exercising or not eating. So the next thing I want to do is put this into some kind of larger context. So I have this big diagram here. So what I'm going to do is I'm going to pull it apart into little pieces so we can understand what's going on. And we want to distinguish the fed from the fasted state. Okay, so let's do fed state first. Okay, so here we are, we're in the liver. And here's the acetyl-CoA carboxylase right here. So if we're in the fed state, then insulin is the hormone that is secreted in the fed state, secreted by the pancreas. So the insulin will activate the PP1, and that will take the phosphate group off of acetyl-CoA carboxylase. So that will make it active. So that means that we can go through our that big fatty acid synthase pathway that I showed you, and we can form palmitate. And again, we'll go to the smoothie R to make other longer or unsaturated fatty acids, and then we'll esterify those fatty acids onto a glycerol, and then we'll get a triacylglycerol. Triacylglycerols are pretty nonpolar. You can't just release those in the bloodstream. They're too insoluble. So we're going to package them up with a little bit of protein and form a very low density lipoprotein particle. And then that goes into the bloodstream, and other tissues can take it up. So that's what happens here, and you can take a whole course just in this, or you can, I teach a nutrition class where we talk about this in more detail, but this is what I'm talking about right here, this part in the liver. So I showed you how the triacylglycerols were converted into the VLDLs, and then those leave the liver and are delivered to the peripheral tissues. And as that happens, the triacylglycerols and some of the cholesterol are removed from them, so these particles become less dense because you're removing lipid from them, but they still have protein. So they become they are converted from very low density lipoprotein to intermediate density lipoprotein and low density lipoprotein. The problem comes if too much of those are around, and then they accumulate in your arteries, and they can cause heart attacks. But if everything goes well, then the role of these lipoproteins is to deliver triacylglycerol and cholesterol to the cells that need it. Okay, still in the fed state. Okay, so so what we have then is here's the VLDL, here's the particle that just arrived, and lipoprotein lipase is an enzyme that is on the surface of the adipocyte, and what happens is the triacylglycerols arrive, and this sounds maybe a little funny, but what happens is the lipoprotein lipase hydrolyzes the free fatty acids in the glycerol, and then it just builds them back up again to the triacylglycerols, and then they're stored in the adipocyte. So the very low density lipoproteins are acted on by the lipoprotein lipase. We get the free fatty acids, and then they're built back up into the triacylglycerols, and there they will stay in our fat cells until we call upon them when we are either starving or exercising. So if we are fed, we don't want to be going through beta oxidation because that we don't need to do that. We don't need to be breaking down fats if we're fed. So this is the what I told you last time, that malonyl-CoA was going to inhibit this carnitine transporter. So remember, malonyl-CoA concentration is going to be pretty high during fatty acid synthesis. So the malonyl-CoA will allosterically inhibit the transporter, and then that ensures that these fatty acids can't get into the mitochondrial matrix to be broken down by beta oxidation. So of course, all of this is hormonally regulated. So now let us turn to the fasted state. And in the fasted state, we have epinephrine that binds to its receptor, and it activates a G protein. So this is very similar to the glucagon activation we talked about before. So the G protein swaps GDP for GDP and activates adenylate cyclase. And then adenylate cyclase catalyzes the formation of cyclic AMP, and cyclic AMP activates protein kinase A. So protein kinase A, what it does is it phosphorylates hormone-sensitive lipase. So again, here we are in the adipocyte, and the phosphorylation of hormone-sensitive lipase by the protein kinase A is going to activate hormone-sensitive lipase. So what it does is it takes these stored triacylglycerols, so these are the triacylglycerols that we made when we were fed. So now we are either exercising or fasting, so we need to call upon them. So we hydrolyze the first fatty acid, then the second fatty acid, and we need a little help from another enzyme right here to hydrolyze the third fatty acid. So now all three of these fatty acids plus the glycerol can leave. And glycerol has 3 carbons, so you can actually ship that to the liver and make glucose out of that. But not any of these guys. Remember, the fatty acids are going to be broken down to acetyl-CoA. To acetyl-CoA, 2 carbons. Can't get glucose out of that. So anyway, so these fatty acids need to leave. But fatty acids, so here are triacylglycerols, there's the phosphorylated hormone-sensitive lipase, phosphorylated by protein kinase A. Here are the free fatty acids, but fatty acids are detergents, so you can't just let them leave because they would cause terrible problems with the rest of the cells in our body if you put a detergent in the bloodstream. So the fatty acids just piggyback on albumin, and then they will go to the liver. So here we are in the liver. So now we're down here. Whoops, sorry about that. I meant to go down here. So here's the fatty acid complex. It's just arrived from the adipocytes. So now the fatty acids are taken up. They're converted to fatty acyl-CoA. The carnitine transporter gets this inside the mitochondrial matrix. So now the transporter is active because there are no malonyl-CoA around to inhibit it. So now we go through beta oxidation. We get a whole pile of acetyl-CoA. It goes to the citric acid cycle if we have OAA. It goes to ketone bodies if we don't. So the ketone bodies, again, are made in the liver, and then they are able to leave for the benefit of other body tissues. So again, the ketone bodies are formed if there's not enough OAA for the citric acid cycle, and that happens if OAA is diverted to gluconeogenesis. So the fasted state summary is here. This is from another book, but it shows this pretty nicely. So here is the hormone epinephrine binding to its receptor, activating the G protein, activating adenylyl cyclase, converting ATP to cyclic AMP, which activates protein kinase A. Protein kinase A phosphorylates hormone-sensitive lipase, and then that converts the triacylglycerols to these free 3 fatty acids. They leave, but they have to be bound to albumin, and then they go through the bloodstream, and then there's a fatty acid transporter. So here it shows not being taken up by the liver, which I showed you on the last slide, but this is being taken up by the muscle. So if you're doing some really long-term marathon type of exercise, your muscles are going to need these fatty acids. So the fatty acids go through beta oxidation. Now here they will go through the citric acid cycle because there's no problem with OAA here because remember, muscles don't do gluconeogenesis, so there's nothing to divert the OAA here. So here there's no problem. The fatty acids go through the citric acid cycle and give you a whole lot of ATP, which you will need when you are exercising. So that was our whirlwind tour of fatty acid metabolism, both catabolism and anabolism. I hope you enjoyed it, and I will see you next time.